Diabetes insipidus.
Jane, a 25-year-old female, was snowboarding without a helmet. While racing down a ski hill, Jane fell and hit her head on a rock. Due to the head trauma, Jane is now diagnosed with central diabetes insipidus.
There are 4 types of diabetes insipidus. Central or neurogenic diabetes insipidus is damage to the neurons in the pituitary gland or hypothalamus, causing a disruption in the production and secretion of the hormone ADH. Causes are head trauma, illness, or neurosurgery. Nephrogenic diabetes insipidus is when the kidneys no longer respond to the ADH hormone. This is an X-linked disorder that mutates the AVPR2 and AQP2 genes. These are responsible for protein production. The causes are genetics, lithium exposure, and/or chronic kidney disorders. Gestational diabetes insipidus only occurs during pregnancy due to an enzyme produced by the placenta that destroys ADH in the mother. Dyspogenic diabetes insipidus is a defect or damage to the thirst mechanism in the hypothalamus. The symptoms of diabetes insipidus are extreme thirst and excessive amounts of urination.
The kidneys are responsible for filtration of waste products in the blood for the production of urine.
ADH movement through the kidneys.
ADH is released from the pituitary gland and goes through the circulatory system. It arrives at the kidneys and travels through the tubules. ADH binds to vasopressin 2 receptors, which are located in the cell membrane of tubule cells. The binding of ADH activates aquaporin 2 water channels to move to the cell membrane. This allows water to flow from the urine back into the blood.
ADH movement with diabetes insipidus.
Due to damaged neurons in the hypothalamus, there is little or no ADH to bind to vasopressin 2 receptors.
Since there is no ADH binding to vasopressin 2 receptors, aquaporin 2 water channels do not move to cell membrane. This prevents water from flowing back into the bloodstream. In regular homeostasis, osmoreceptors detect a change in osmolarity of the blood. They then send a message to the hypothalamus, which produces ADH. This is then sent to the pituitary gland and moved to the kidneys, where it is used in processes stated before to maintain balance and osmolarity of the blood. In central or neurogenic diabetes insipidus, the ADH is not being produced or delivered due to damaged neurons. This prevents osmolarity from being balanced in the blood.
Treatments include desmopressin, which is a hormone replacement for vasopressin, which helps reduce urine production. Jane can also manage her symptoms by cutting down on the amount of water she drinks to control excessive urination.
In summary,
proteins and cell membranes. Osmoreceptors continue to detect osmolarity, but ADH can no longer be produced. The production and secretion of ADH becomes disrupted due to the damage of neurons in the hypothalamus. Vasopressin 2 receptors no longer receive ADH due to the lack of production and secretion of this hormone. Aquaporin 2 water channels are no longer stimulated, so water cannot flow from urine to blood. Vasopressin 2, aquaporin 2, and osmoreceptors are all embedded in the cell membrane.
homeostasis. In neurogenic or central diabetes insipidus, the neurons become damaged and can no longer produce, store, or transport ADH to the kidneys, causing an excessive amount of urination due to the lack of ADH stimulating aquaporin 2 water channels.